My name is Christian Fercher, and I'm the operation manager of the protein expression facility here at the University of Queensland, which is a fairly large university on the east coast of Australia in Brisbane. The protein expression facility, or as we call it, the PEF, is part of the University of Queensland as a core facility. And we sort of specialize in producing recombinant proteins to support a, really wide range of research activities. And that includes everything from early stage fundamental science through to translational and preclinical projects, essentially. In terms of our clients, we work with pretty much the whole research community across the university, across other universities in Australia and even globally, with government partners and industry clients. I would say beyond expression, we have a fairly deep capability in purification and also analytical characterization of the final products or intermediate products that came out of stability studies. Before the Maurice came into our lab, we mostly relied on traditional SDS paged gels for evaluating the purity and the sort of homogeneity of samples. And then if you look at the isoelectric focusing capability, we employed analytical cation exchange chromatography to analyze the charge variance. In terms of where the Maurice fits in, it actually fits into multiple stages of this process. Like the early screening that really helps us quickly to assess how different constructs behave. They also give us a very good idea about things like the purity, the charge distribution, the isoelectric point. Maurice really gave us the ability to expand our analytical framework and to look at these sort of things very quickly without necessarily having to optimize a analytical workflow on an HPLC-based method, or potentially even having to outsource these measurements and then waiting for the results for much longer. That this is possible when you have an instrument in-house. As a specific example, we had a project that was involving a highly glycosylated fusion protein from a higher eukaryotic organism. And on SDS page gel, it looked like fairly clean, maybe one or two variants at max, a bit of a smear, as you would generally expect to see. But once we ran this on the Maurice, we saw a much more detailed distribution of very subtle variants that were completely masked on the gel and were absolutely invisible by this technique. And that really helped the team to refine the purification strategy for once, just because, we knew the exact isoelectric point now. We see approximately 150 different research projects every year. That means I see 150 different proteins a year. And I think it's such a phenomenal throughput and such an experience to see how they all behave differently, what makes a really good purification platform.